History of DNA arrays. Grunston and Hognes created the DNA array with the colony hybridization method. They randomly cloned the DNA of interest into E. coli plasmids that were plated onto agar Petri plates covered with nitrocellulose filters. Replica plating was used to produce additional agar plates. The colonies on the filters were then lysed and their DNAs were denatured and fixed to the filter to produce a random and unordered collection of DNA spots that represented the cloned fragments. Hybridization of a radio-labeled probe of a DNA or RNA of interest was used to rapidly screen thousands of colonies to identify clones containing DNA that was complementary to the probe. This approach was adapted to create ordered arrays by Jergen in 1979, who picked colonies into 144 well microplates. They created a mechanical 144 pin device and a jig that allowed them to replicate multiple microtiter plates on agar and produce arrays of 1728 different colonies in a 26 by 38 centimeter region. An additional transfer of colonies to the squares of Whatman filter paper, followed by a growth, lysis, denaturation and fixing of the DNA to the Whatman filter paper, allowed the production of DNA. During the next decade, filter based arrays and protocols similar to these were used in a variety of applications, including identifying SNPs, cloning genes of specific interest, cloning genes that are differentially expressed between the two samples, and physical mapping. Hans Leyrag's groups automated these processes in the late 1980s and early 1990s by using robotic systems to rapidly array clones from microtiter plates onto filters. The concomitant development of cDNA cloning in these periods, combined with international programs to fully sequence both the human transcriptome and human genome, led to create reference sets of cDNAs and cDNA filter arrays for human and other genomes. By the late 1990s and 2000s, sets of non-redundant cDNAs became widely available, and the complete genome sequences of some organisms allowed for sets of PCR products representing all the non-open reading frames in small genomes. These sets, combined with readily available robotics, allowed individual labs to make their own cDNA or ORF arrays that containing gene content represented the vast majority of genes in a genome. The birth of the modern DNA array. In the late 90s and 2000s, DNA array technology progressed rapidly as new methods of both production and fluorescent detection were adapted to the task. The knowledge of the DNA sequences of multiple genomes provided the raw information necessary to assure that arrays could be made which fully represented the genes in a genome, all the sequence in a genome, or a large fraction of the sequence variation in a genome. A gradual transition from spotting relatively long DNAs on arrays to producing arrays using 25 to 60 base pair oligos was noted during this time. The transition of oligo arrays was made possible by the increasing amounts of DNA sequence information. The use of oligos as opposed to longer sequences also provided an increase in specificity, for the intended binding target as oligos could be designed to target regions of genes or the genome that were most dissimilar from other genes or regions. Three basic types of arrays came into play during this time frame: spotted arrays on glass, in situ synthesized arrays, and self assembled arrays as shown in figure 2. Spotted arrays. In spotted arrays, polylysine coated glass microscope slides provided good binding of the DNA, and a robotic spotter was designed to spot multiple glass slide arrays from DNA stored in microtiter dishes. By using slotted pins similar to the fountain pens in design, a single dip of a pin in DNA solution could spot multiple slides as shown in figure 1. Spotting onto glass allowed one to fluorescently label the sample. Fluorescent detection provided several advantages relative to the radioactive or chemiluminescent labels common to filter based arrays. First, fluorescent detection is quite sensitive and has a fairly large dynamic range. Second, fluorescent labeling is generally less expensive and less complicated than radioactive or chemiluminescent labeling. Third, fluorescent labeling allowed one to label two or more potential samples in different colors and co-hybridize the samples in the same array. As it is very difficult to reproducibly produce spotted arrays, comparisons of individually hybridized samples to ostensibly identical arrays would result in false differences due to array variation. However, a two-color approach in which the ratio of the signals on the same array is measured is much more reproducible. In-situ synthesized arrays. In-situ synthesized arrays involves chemical synthesis which combined photolabile protecting groups with photolithography to perform chemical synthesis on a solid substrate. In this production of arrays of 10 amino acid peptides and separately arrays of dinucleotides was demonstrated. Affymetrix demonstrated the ability to use this technology to generate DNA arrays consisting of 256 different octanucleotides. Affymetrix arrays are being used to detect mutations in the reverse transcriptase and protease genes of the highly polymorphic HIV-1 genome and to measure variation in the human mitochondrial genome by 1995 to 1996. Eventually, Affymetrix used this technology to develop a wide catalog of DNA arrays for use in expression analysis, genotyping and sequencing. A major advantage of the Affymetrix technology is that the DNA sequences are directly synthesized on the surface and only a small collection of reagents, that is the 4 modified nucleotides plus a small handful of reagents necessary for the deblocking and coupling steps are needed to construct an arbitrarily complex array. A method proposed by Blanchard and his co-workers in 1996 used inkjet printing technology and standard oligosynthesis chemistry to produce oligo arrays. In this method, inkjet printer heads were adapted to deliver to the 4 different nucleotide phosphoradamides to a glass slide that were pre-patterned to contain regions containing hydrophilic regions with exposed hydroxyl groups surrounded by hydrophobic regions as shown in figure 1. The hydroxylated regions provided a surface to which the phosphoramides could couple, while the surrounding hydrophobic regions contained the droplets emitted by the inkjets to define regions. This technology was eventually commercialized by Rosetta Informatics and licensed to Agilent Technologies, who produce these arrays at present. The inkjet array approach shares the advantage of Affymetrix and Nimblegen approach in that one only needs to have a small number of reagents to produce an array. In addition, similar to the Nimblegen approach, the production of a new type of array only requires that a different set of sequence information is delivered to the printer. Hence, the inkjet array technology has been particularly useful for the design of custom arrays that are produced in low volume. Self-assembled arrays. An alternative approach to the construction of arrays was created by the group of David Walt at Tufts University and ultimately licensed to Illumina. Their method involved synthesizing DNA on small polystyrene beads and depositing those beads at the end of the fiber optic array, in which the ends of the fibers were etched to provide a well that is slightly larger than one bead. Different types of DNA would be synthesized on different beads, and applying a mixture of beads to the fiber optic cable would result in a randomly assembled array. In early versions of these arrays, the beads were optically encoded with different fluorophore combinations in order to allow one to determine which oligo was in which position on the array, referred to as the decoding of the array. Optical decoding by fluorescent labeling limited the total number of unique beads that could be distinguished. Hence, the later and present day methods for decoding the beads involve hybridizing and detecting a number of short fluorescently labeled oligos in a sequential series of steps.